this year as a part of strengthening the bonds between alma mater and the alumni. That is one aspect. Second thing, when the students know that the alumni are in very good position spread throughout the world, that will be a motivation to the students. Third, students can also come in contact with the alumni, and whenever they have some doubts regarding what career they have to select, what they have to do in the future, they can have guidance from the alumni who are already excelling in their profession. So with these three intentions, we started this alumni lecture series in which every month one alumni who is in different parts of the globe will be called here to deliver a lecture in the field of their expertise so that the students will be in touch with them. So now today we have Ms. Shruti, who is well settled in Australia and excelling in her profession in the regulatory affairs. So Professor Subramaniam sir thought of calling her so that it will be a great motivation to all our students. Now I would like to welcome all again, and I'd like to hand over the stage to Professor C.V.S. Subramaniam for the introduction of the speaker and to start the further proceedings. Sir.
Thank you very much, sir. Good morning, every one of you. I take this opportunity to welcome Shruti Challa on behalf of the students, staff, and the management to speak on this particular platform, Alumni Series. It is a privilege to introduce her to the students of PG students as well as some new staff members and also some staff from other colleges also. Shruti Challa graduated B Pharmacy with distinction in 2008. She is the second batch outgoing student of our college. Immediately after B Pharm, she was offered a job in management training in Dr. Reddy Laboratories. During her course, she had a training in aseptic production of snake venom anti-serum production and QA in Hyderabad. After this, she had migrated to Australia. At present, she is Regulatory Affairs Manager, RA Manager, Grifflows Australia, Private Limited Australia. It is actually in Victoria, that is very south to Australia continent. It is New South Wales, and at least it is below the Sydney. That means she is very near to New Zealand rather than Australia, I think. After she going from India, she has mastered in GMP in pharmaceuticals with Swinburne University, Australia, with honors. And in fact, in Australia, the GMP comes under QA, and pharmaceuticals as well as medical devices are combined together, and they call it as therapeutic goods administration, that is TGA, thermogravimetric analysis. So all these are combined, and she had specialized in that particular post-graduation. She did diploma in leadership and management also as a fast-track course by way of handling her job, coaching, mentoring, and managing people, undertaking project work with others, etc. That is how she has got a multifaceted activities. Earlier, she worked in Surgi Supplies as medical devices manufacturing company, and she also, and during that particular time, she is responsible for developing and various profiles, applications, etc., to be submitted to the, whether it is USA or Europe or Australia or Canada. Like that, several organizations she had applications, and for some time she was also consultant in the RA. Particularly, I hope that she has got a very good grip in medical devices activity which normally we are not very much familiar with in India, right, madam? So she has publications, she has publishes research articles, and she has written a book also, and several activities that are concerned with a company and employees, etc. And that is how she has been a consultant and also a guide or a judge for various activities concerned with RA, etc. So she is considered to be a very proud to have Shruti madam, such a high profession and doing extremely well in Australia, and who is an ambassador for GRCP, Hyderabad. Thank you very much for accepting our invitation, madam. With these remarks, I hand over the mic to you, Shruti, to take care of, address our staff and students. Thank you very much.
Sure. Thank you very much, Subramanian sir, for the great introduction. And you don't need to call me madam, you can call me Shruti. That's sufficient. Small correction, sir. I didn't do any book. I didn't publish any book. I'm not as great as you. I've just done an article, but I haven't done any book. I haven't written any book, or I haven't published any book. But rest everything
that is technical writing. Technical writing.
It's just it's a course I have done in technical writing, but yeah, I haven't done any book. Thank you very much all for coming here. It's a pleasure and honor to be here to present this lecture to you all, because I am an ex-student of GRCP. I passed out in 2008, and I moved to Australia, and now I'm working as a regulatory affairs manager in Grifols Australia, which is a pharmaceutical as well as a medical device manufacturing multinational company. I'd like to introduce you to some concepts about validation and help you understand how validation activities fit in pharmaceutical manufacturing as well as medical device, but particular emphasis on pharmaceutical manufacturing alone. I'll start the PowerPoint presentation now. I will present my screen. Okay. I hope you see the screen. Could you please let me know if you could see the screen?
Yes, madam, you can continue. Your presentation is started.
Okay. Thank you very much, sir. So, yeah, the topic today I'm going to present a lecture on is validation of process and equipment. A brief overview. It is a really big concept. We cannot go through the entire details of validation in this 1 hour, but we'll try to go just the brief concept of validation and how it fits into process and equipment in pharmaceutical manufacturing companies. The reason I have chosen this particular topic is I sort of struggled to understand the concepts of validation as I was a student, but when I moved into industry, when I started working in the industry, I realized how important validation is. And without validation, we cannot release the product. So validation has got a supreme importance in pharmaceutical manufacturing industry. So to understand the validation, we'll first see where the validation fit in pharmaceutical manufacturing. As you all aware, I suppose, to manufacture pharmaceuticals, we need to comply with regulatory requirements. For example, regulatory requirements in India for pharmaceutical manufacturing is Drugs and Cosmetics Act 1940, Schedule M. In Australia, the governing body is TGA, and it's called Good Manufacturing Practices. In USA, the governing body is FDA, and the regulatory requirements for pharmaceuticals is 21 CFR 211. In European regulations, it's again called Good Manufacturing Practices. They have regulatory requirements in the form of directives. So it's Directive 91/412/EEC is the directive for pharmaceutical manufacturing. Good manufacturing practices are applied in several different areas in manufacturing of pharmaceuticals. It's broadly classified into production and quality control, where good manufacturing practices are applied. In production, we need to monitor and control suppliers, materials, facility and equipment, various processes, non-conformances, corrective action, preventive action, and customer complaints. So our validation would broadly fit into these two areas, which is processes, facility, and equipment. So it is a GMP requirement that all the critical steps in manufacturing and quality control are validated. When I say critical steps, it could be most important steps which would affect the quality of product. For example, if you look at tableting, granulation is an important step which would affect the final quality of the product. So that is considered as a critical step. Tableting process is a critical step. In that way, we have several critical steps in manufacturing of a particular pharmaceutical product. Every single critical step needs to be validated. That is a GMP requirement. And critical equipment, which means the equipment which has impact on the final product, also need to be validated. When I say critical equipment, it also extends to facility as well as systems. So in the last section of the slide, I've given examples of critical steps, as I just spoke before, like granulation, mixing, blending, compression, etc., and process of manufacturing of tablets. So what is process validation? We'll see the definitions from FDA. When validation involves establishing a documented evidence which provides a high degree of assurance that a specific process will consistently produce a product meeting its predetermined specifications of quality attributes. Please focus your emphasis on consistently producing a product meeting its specification, which means if we are taking any critical processes, for example, mixing. So we'd have validation is something which we consistently ensure that mixing process results in having a uniform blend mixture. And this particular activity, validation, needs to be documented. I hope you'd understand. Please go through the definitions a few times so you'd get more knowledge for the students. So in validation, there will be critical process parameters and critical quality attributes. A critical process parameter is a parameter whose variability has an impact on critical attribute and therefore should be monitored or controlled in order to ensure that a particular process produces the desired quality. CQA is physical, chemical, or biological or microbiological property or characteristic which needs to be in a particular limit, range, or distribution in order to ensure desired product quality. We'll look at some examples of critical process parameters and critical quality attributes, CPPs and CQAs, to understand better. Please look at this process. In sieving process of manufacturing tablets, the critical process would be sieve diameter and raw material grade. These parameters are variable, which means the sieve diameter can change, can vary. It's called variable. But this critical parameter would affect the quality attributes. If the sieve diameter changes, it would affect the particle size distribution. It would affect the bulk density. So in validation, we would actually validate the sieve diameter and we will determine which sieve diameter is the best to achieve the particle size distribution. We'll look at another example. The other example is granulation. Here the CPPs would be blend speed, volume of the fluid, feed rate, and CQA is the granule uniformity. So these are the variables: blend speed, volume of the fluid, and feed rate are the variables affecting the granule uniformity, which is the quality attribute of this particular process. So in order to understand this CPP and CQA, please emphasize your focus on quality. If you say it's quality in granulation process, you ensure the quality having a granule uniformity. We'll go to another. If you look at the process of tableting, what could be the CPPs and CQAs? I'll tell you on the next slide. If you can make a guess, you can make a guess. CPP is a compression force and excipients, and CQAs are disintegration and hardness. So these two CPPs, compression force and excipients, would affect the critical quality attributes, which is hardness and disintegration. I have given a sample process validation protocol for mixing. Mixing is the process here. So a protocol gives the plan of how we are going to validate the process of mixing. So for any protocol, the aim is important, aim or purpose, we need to document. Here, the aim is we have taken a chlorine phosphate syrup, for example, just for an example's sake, on how to do process validation for this particular thing, to consistently demonstrate homogeneity of final bulk mixture of chlorine phosphate syrup before filtration or packaging. So the purpose of this process validation, what we're going to do is to demonstrate the homogeneity of final bulk mixture. We have got chlorine phosphate syrup. We want to demonstrate every time we do mixing, it gives the homogeneous chlorine phosphate syrup. So this is the aim of this particular process validation. Experimental runs. This is a standard requirement from FDA and also from PGA that we need to do at least 3 batches for conducting process validation. So we've taken minimum runs as 3. We need to choose an equipment. That's a mixing tank, 600 liters of mixing tank. It's an example. What are the critical process parameters for this mixing step? It's mixing speed and time. So here in this case, we are keeping time as constant, which is 60 minutes, and mixing speed, we can change. It's a high speed or low speed. There are 2 different settings for this mixing tank. Just consider for this case sake. There are 2 speeds, high speed and low speed. So we are going to validate the speed, mixing speed, to ensure that every time we do proper mixing to do a homogeneous mixture. We're keeping time as constant here. Experimental details, how it's going to be. For first run, we said in the previous slide, we're going to take 3 batches, 3 runs. And the first run, we'd keep the speed setting as low. And the second run, we'd keep the speed setting as high. And run 3, we can keep the speed setting high or low, depending upon the result of first and second runs. For example, if we get a homogeneous mixture in run 1 with low speed, so we will ensure even the run 3 is low speed. So we'd have more assurance, okay, low speed, the first run we got a homogeneous mixture, and also in run 3 we got a homogeneous mixture. So let's go for low speed, low mixing speed. So depending upon this, we do the run 3. And we need to have some sort of a sampling plan in place to identify the homogeneity of the mixture. So we do sample at 15 minutes, 30 minutes, 45 minutes, 60 minutes. We take 2 samples each time from top of the tank and bottom of the tank. And all these things in industrial space, you have procedures for each and every activity. So we are assuming there is a standard operating procedure for sampling plan. If there is one, we would give reference saying, okay, follow this sampling procedure, SOP, so on. So once this is done, we would check the performance parameter, and we do assay of the sample. We'll check whether the sample is homogeneous or not by checking the pH, density, ethanol content, and methyl paraben. We see the quantity in the sample mixtures to see how the homogeneity is there, whether this in this run with having high speed, whether we have all these things meeting the specifications or not. So the acceptance criteria would be the specifications of all these: pH, density, ethanol content, and are our test meeting the specifications or not. If run one fails, okay, the second one, is it meeting the specification or not? So that's what we need to record. So once the protocol is done, process validation protocol is done, we would do this experiment or do this validation exercise for 3 runs, and we do document the record. This is still the process validation protocol continuation. I just included this slide for helping you to understand the importance of signatures. For any document, we need to have signatures, author's name and signatures in place. All the documents need to have at least one person reviewing and approving. And date is also very important. This has the full set of different regulations. It's called good documentation practices. In industry, we need to employ good documentation practices. If there is no date, the auditors may come and look at the facility, and they would ask when you have done this protocol. Is it after you have finished the validation or before you have to finish the validation? If we have done after the validation, then the validation is not good. So dates have such great importance in industry, and also signatures. So when you go out into the industry, please ensure that you're extremely careful in documentation, because you may carry out any activity, but if you don't document, which means it's not done. The auditor cannot verify you have done an activity or not, and they would say, no, you haven't done. That's all. So be extremely careful in doing documentation. We'll go to the next slide. This is about process validation protocol. This would result in the experiment or validation exercise, and also the record, which I haven't included in this presentation. But that will follow with the record. So we'll go to the next topic, which is equipment validation. In equipment validation, this is the model from FDA. It is called V model. It will start with user requirement specification. Sorry, is there any interruption? Sorry, okay. This is called V model from FDA. This have all these elements we need to carry out for doing equipment validation. We need to carry out validation for all the critical equipment. And I say critical, any equipment which impacts the quality of the final product is called critical. So, for example, we want to buy a mixing equipment. We'll start with a user requirement specification. This is usually done by the manufacturer. That will list out what the user wants in user requirement specification. So the manufacturer would document a document saying, I want this mixing tank, which is of 600 liters, for example, and it needs to have settings of this setting to that setting, temperature setting, speed setting. So and he would give a list of requirements to the vendor, and he would approach to the vendor. The vendor would match, giving his details matching with the user requirement. It's called functional specification. The vendor would get back and say, okay, I've got a mixing vessel which is of 600 ml 600 liter capacity, and whatever you want, we can match. So he would create a document matching to the user requirement specification, giving all the specifications which matches with the user requirements. And the vendor would also provide the design specification. We asked for a equipment, so he'd give the detailed design of how the equipment would look like, what the cost, the details of all the design equipment, the design specifications he would be giving. So once we buy the equipment, we'd get it into the facility, pharmaceutical manufacturing facility, and we need to carry out these 3 activities. We need to install it correctly, we need to operate it, and we need to perform process qualification to ensure that particular equipment is good for doing whatever activity we want. For example, that mixing vessel what we purchased is installed correctly, it operates correctly, and every time it can give a real good result. It can mix effectively. In simple equipments, the process qualification and the performance validation would overlap. For example, because the mixing equipment is not a high-level equipment, it's a simple equipment. So we install, operate, and also do the process qualifications or performance validation would be overlapping. But if you look at a really big equipments, then it would have a separate process qualification, and it would have a separate process validation and separate process validation and separate process qualification. Okay. So what I have told you, I have included here. User requirement specification, it's a formal document of what users want. For example, mixing tank we want with so-and-so capacity, operating range from so-and-so, etc. Functional specification is provided by the vendor, and detailed design specification is also provided by the vendor, which maps directly to the user requirement specification. So these are all the documents associated. Then once we get the equipment, we would install. We do installation qualification, we do operational qualification, and we do performance qualification. Installation qualification is a documented verification that a particular equipment is installed to comply with the design and manufacturer's recommendations. Operational qualification is a documented verification that a particular equipment function as intended throughout the operation range. Performance qualification is a documented verification that a particular equipment can perform effectively and consistently to meet the product specification. We'll look at an example now. To have an easier understanding, I have taken pedestal foot sealer. This is a very simple equipment. It is a heat sealer equipment where we put packaging in between these 2 jaws and we hit the pedestal, and that would cling both the jaws and that would seal the packaging. It would have a temperature probe which is calibrated, and it has an operating range, and we can adjust it depending upon the type of packaging we want to seal. Okay, so we bought this equipment. We did all the URS, and we got the functional specification from the vendor and design specification. So we got it, and we want to install. So we perform this installation qualification. So what does this installation qualification include? Because this is such a simple equipment, it doesn't have much for installation qualification. All we are checking here is, because we said we got a temperature probe, we need to have it calibrated. So is the sealer one calibrated? Yes or no? Is the sealer surface free from cracks or any other visible damages? Because if there is any damage to the sealer, it doesn't seal properly. We are checking for that. We need to be trained before we use the sealers, trained on the safety. So we are checking for these 2. And the sealers have to be kept under preventative maintenance. In pharmaceutical equipment, in pharmaceutical space, it's one of the GMP requirement that the critical equipment need to be kept under preventative maintenance. Preventative maintenance is the routine maintenance, like It's done every twice in a year. It's a routine. You do the preventative maintenance before the sealer breaks down, before anything happens to the sealer. The idea is we do preventative maintenance before the sealer breakdown, because all of a sudden if sealer breaks down, we cannot carry out the sealing activity. So the production would be interrupted. So we don't want that to happen. So we would have that included in preventive maintenance schedule. And every year we would do the preventative maintenance. For this equipment, the maintenance is as simple as just oiling the jaws, ensuring the jaws go well, and doing calibration of the temperatures, temperature probes, etc. routinely every year. So we check for the power output, and we'll check for the training. That's all we do for this particular equipment. The complexity of installation qualification increases as the equipment becomes more complicated. And this activity would require inputs from engineers, inputs from quality assurance people. They need to give what is required for proper installation of the equipment. Next, we'd go to operational qualification. We install the sealer. We just want to check whether it operates, that's all. So we turn it on and off. Does it turn on? Does it turn on when turned on? Yes or no? That's it. Does its jaws open and close using the foot pedal? That's all we are checking for. So, and we have written comments here. Yes, the switch is turned and the seal surface gets heated up. The switch is turned off, the seal surface gets cooled down. And this is the acceptance criteria. And we have checked and have written. Jaws on applying the pressure on the foot pedal. Jaws open and close. Jaws open and close, and we have checked for that. So this is operational qualification.
No, I can't ask that. This is not this webinar.
Sorry, is there an interruption, sir?
Madam, continue. You continue, madam.
Oh, okay. All right. So we'll continue with performance qualification. We have done the installation qualification, operational qualification, and we'll check for the performance qualification. Because this is such a simple equipment, which is a foot sealer, we don't need elaborate process validation. The process validation and performance qualification is same in this instance. But if we go to a really complicated equipment, for example, sterilization equipment, for example, a chamber which does sterilization of pharmaceuticals, for example, that would have a different pharmaceutical qualification. Sorry, that would have a different performance qualification. In that instance, the performance qualification would include connecting to different utilities, just doing a sample trial runs, whether it's performing as intended or not, and checking for temperatures, pressures, etc. And the process validation would include the actual pharmaceutical product would be placed inside the sterilization chamber, and we are checking whether our sterilization chamber is good enough to sterilize the pharmaceutical product we are putting in that or not. So that would become process validation. So because this is such a simple equipment, as I told you before, we haven't done a separate performance qualification. We combined along with the process validation. We did have 3 different types of packaging, which we wanted to seal using the sealer, which is Steriking, Steripack, and Tyvek. These are 3 different types of samples or packaging, what we wanted to seal using the sealer. So we need to take some samples or packaging based on statistically valid rationale to do this performance qualification. So based on the statistically valid rationale, we have taken 60 samples of each type, and we seal using the sealer, and we do the test to check whether the sealer is good or not. There are two different tests here, visual inspection and dye penetration. We check all the samples for these two tests, and we check whether it's good or not, and they have passed or not before we start using it for sealing the actual devices. Actually, this particular equipment we use for sealing of sterile medical devices. I have taken this because it's a very simple example and help you understand the equipment validation. So the two tests we conduct here is dye penetration test. In order to ensure the seal is done properly or not, we include a little bit of dye. We inject a bit of dye using a syringe, and we let the dye go into the packaging. If the packaging is not sealed properly, it would cause the dye to leak. And if the dye leaks, the packaging has failed. So that's one test. There is a visual peel test where we just open the package and see once it's been sealed to see if there are no cracks, no crevices, and the seal is properly laid or not. By just looking at the packaging after sealing, we can identify whether the sealing is done well or not. So these are the two tests we conducted. So this is the record. In the previous example of process validation, I have included the protocol, and this time I have included the record. So this is the sample record. We have taken the samples. Number of samples would be 15 samples. The temperature would be, we are checking at 2 different points. Oh, this is still, okay. 2 different points at 195 degrees and also 200 degrees. We have taken these two as the operating range of the sealer. And we do check for visual inspection as well as dye penetration. So 15 samples of steriking at 195 degrees. We do visual inspection. 15 samples, we do dye penetration. 15 samples at 200 range, we do visual inspection, and 15 samples for dye penetration. So altogether 15, 15, 15, 15 will be 60 samples. In that way, we have done for 3 different packaging, and we are checking for both lower temperature and also high temperature. And we do those 2 tests, visual inspection and dye penetration. And depending upon the pass and fail, we'd set up the temperature routinely. If the sealing is good at 200 degrees temperature, we'd operate the sealer at 200. The performance qualification record looks like this. So that includes date, the sample number, temperature, and evidence of leakage in dye penetration test, and samples and evidence of leakage. So this is exclusively for the dye penetration test. Yep, that's it. So that's about equipment validation. So I've just included the consequences for not doing validation. Validation has got a great deal of importance in Australia and also in USA, in pharmaceutical manufacturing companies and also medical device manufacturing companies. The idea of validation is we need to ensure that a particular process is done correctly and it will give a product which is of good quality. Because if we just do quality control tests at the end of production, we'd be sampling only very few and we'd be conducting the quality control tests on them. So the quality control test may pass, but if we don't validate, you never know. All the other products which are already in the market may fail. So when we do the validation, we are ensuring that all the product we are putting into the market have got good quality. So the idea here is assuring the quality. Quality shall be built into the product, not just being tested in the end product. So validation has got a great deal of importance. And it also saves money for the manufacturer. And also, for example, I can give an example about the mixing. mixing of the codeine syrup, for example. We haven't validated the mixing speed, but we started manufacturing using the mixing speed of high, and we have done 600 liters of codeine syrup. Finally, we have got some samples out and we did quality control tests. Imagine the quality control tests have failed. What we would do? We need to quarantine the whole samples and we need to dispose, which means throw away. It's going to be a very costly affair because we need to get raw materials, we need to run the equipment, we need to provide salaries for the people working. So it's going to be very expensive. But if we validate the process beforehand itself, every time we do lots and lots of batches, we would have high level of confidence that we would pass the quality control test. So that is the idea of validation. So that's applicable obviously to process as well as equipment. If we have a good equipment and it's properly installed and it operates correctly and it performs as intended, we don't have any issues, we can keep on running it. If an equipment breaks down in big manufacturing facility, the production would be interrupted. That would be very expensive. So that's why we need to have that equipment placed in preventive maintenance schedule, and we need to routinely maintain it before it breaks down. So those are the concepts of validation. So what are the consequences of not doing validation? In FDA, if the FDA auditors come and find out that we haven't validated a particular process, they will issue a warning letter. If it is a critical equipment that we haven't validated or critical process we haven't validated, like for example, the sterilization chamber, it's not been validated and we are supplying sterile products, they would even suspend the license, and also they would ask us to recall the entire product, because we cannot assure, or we don't have high level of confidence that all the products, sterile products we placed into the market are sterile. We would have done the quality control test, like we would have taken a couple of samples and we have done a sterility test on them, but that's not sufficient. Civil penalties and criminal penalties happens if the person is being given a non-sterile sample. We haven't validated the process. Our process is not good, but we have distributed the sterile pharmaceuticals and something happens to the person, then there would be civil penalties. And if the person dies, there will be criminal prosecutions. So the validation has got a great deal of importance. I work in pharmaceutical company at the moment, pharmaceutical and medical device, where I do regulatory submissions to TGA. And every other day, if there is a change in pharmaceutical process, we need to submit a notification saying, we have changed so-and-so process, and we have done the validation, please see. And they're gonna check the validation study, and they're gonna approve, okay, that's fine, you can go ahead with the change. Similarly, the same regulation also applies for medical devices. If there is any change to the medical device, which we think it's gonna impact the final product, we need to do the validation study, and we need to notify to TGA saying that we have changed so-and-so process, and we have conducted validation, and please see the documentation. They would approve and they would ask us to go ahead with the change. Only then we can go ahead with the change. So this is the end of lecture on validation. Thank you very much. I would like to still continue talking. I just would like to talk casually, like a general talk, general topics and personal experiences. We are in COVID situation in India at the moment, which is a very serious situation. So please, everybody, be extremely careful and reduce your socializing activities. It's not worth taking any risk. The whole life is ahead of you. Because I come from your age, I'm addressing to the students of GRCP, where I used to like to socialize at your age. Go ahead, go and meet friends and go out. But at this serious situation, it's not worth. So please be safe. I'd like to share a few other suggestions which are career related. The importance of communication and importance of networking. Please develop your communication skills, which includes oral and written. English being a second language, it requires more practice. So talk more, write more, read more, watch and listen to English more, and even think in English. Because we have a different native language, and to speak in English, we think in our native language, and we translate in English and we talk. So there would be a little time lag. So it requires a lot of practice to come natural flow, to have a natural flow in talking in English. And it has a lot of importance in our career, because we might have lots of ideas, but if you're not able to communicate it properly, it's of no use. So focus quite a lot on improving your communication, and also written communication, emails, etc. If you're doing it professionally, if you're writing an email to your professor, lecturer, please have good English. Use your capital letters properly, punctuation, grammar, everything properly. Importance of networking. I want to emphasize an importance of networking, because it's quite important to network with people to get the real situation in the industry. LinkedIn is a very good platform to connect with people. Reach out for recruiters, talk to people, talk to your seniors, so you can get an idea of what's going on in the industry and what you need to do in order to advance in your career. So have a good LinkedIn profile and start networking with people. The final suggestion I would say is just believe in yourself. Don't have the fear of failure. Be bold to take a step. Don't procrastinate. If you want to do something, if you want to do something in your career, don't be lazy. Don't keep it away. Just work and get it done. It's a very competitive world, but you can still succeed if you believe in yourself. Be aware of negative people around you. I'm not kidding, but I myself have experienced having negative people around me. When I finished my master's here, I had housemates who were telling, there is no chance I'm going to get a job in Australia. There is no chance. Not even an internship. And they strongly pushed me to go back to India and get a career in India, stating that I would not get a job, because getting jobs in Australia is so difficult for life sciences group. That's the rationale. It was a bit hard to find a job. It took nearly 6 months, but I could get a job in industry, and I'm in very good position than many of my fellow beings or the people, my housemates, who has negatively told about getting jobs in Australia. Get the best out of you. Gain more knowledge. Present yourself well. Your way of conducting yourself, because all this matters in getting your job and also making a career, because people are pretty judgmental. It's not just about grading grades. The way you talk, the way you conduct yourself, the way you present yourself matters quite a lot in your career. Finally, just be a hero of your life. Take charge of your personal and professional life, because in life you face a lot of challenges, and you need to strive and learn and take charge of your personal and professional life. Thank you very much for your time. I finish my lecture here. I hope I didn't bore you. If you have any questions, you can feel free to ask. I'd also say thank you.
Any questions?
Yes, sorry. I'll go with questions first. Yeah. Sorry, I can't hear you, sir.
This is
Sir,
Kumar is asking, is design qualifications comes under FAT, process analytical techniques? I think it is PAT, I think.
Or they could be validation also in analytical procedures, and also non-analytical procedures. In just the equipment itself, there are design specifications which we need to which we need to get from vendor.
Cool.
Yes, the GMP or validation principles are not only applicable to process equipment in production, but it's also applicable in quality control. I hope you have answered.
Under broad heading of RA, that is regulatory affairs, one PG course you have mentioned as GMP.
Yes, sir. Yeah, that's my master's.
Specializations are there?
Sorry?
How many more specializations are there? Under regulatory affairs, RA.
GMP, I am a quality assurance and regulatory affairs professional, sir. In quality assurance, deals more with GMP, where I need to implement a quality system, where I need to do a lot of documentation, write procedures, train people, managing CAPA, managing corrective actions, preventive actions, and complaints. That's one side, which I had worked in my previous companies. As a regulatory affairs, now at the moment, my job is doing registrations with TJ. So Grifols is a big company. It has many branches in Spain, USA, but every single product what we put into Australia needs to go through TJ. So my job is getting the information from different areas of the world in Grifols and compiling, and I need to be well with the regulations of TJ. So I need to ensure all the documentation I have received, and I need to put it to TJ and communicate with TJ and get approvals, and that's my job now. If I was into quality assurance, I would be a quality assurance manager. I would do procedures. I need to train people. I need to face audits. I faced a lot of audits with TJ in my previous companies, but that's not the job I'm doing. I'm doing the regulatory affairs now. I hope I have answered. Yes, sir?
Yeah, Kangaraj, you want to speak?
No. You get, no.
See, because of this particular validation and all these procedures, how much time the life cycle, product life cycle has been increased? How much time? Is it 10% of the product life cycle is meant for only these validation and qualifications, or 15% or 20%?
I cannot say the exact percentage, sir. It depends upon the process we are implementing in our company and how complicated the process is. For example, I have given an equipment validation. I have done it in a week. I have done the protocol. I have done the record. I have done the testing in a week. But we have the sterilization process in place to do the sterile medical devices, which is a really complicated process where we use a oxide chamber for doing the sterilization process. So there is a great deal of validation there. So we need to validate the chamber. We need to do performance validation of the chamber every year. So every year, 1 month would be spent for validation.
Yeah.
And it's also an expensive exercise. We need to do a duplicate run. We need to send some samples out to the sterilization, get some samples out, and also do the sterility test. We need to do the shelf life. So because of the complexity of the process, it would take more time. So in big companies for manufacturing sterile products, of course, it would take a long time. But if they're not manufacturing sterile products, it may not take such a long time. Yeah.
Any more questions, please? You can ask, unmute and ask by the participants. In fact, your presentation is very good, madam. It has changed my mind to only.
Madam.
I'm trying to get ideas how to present it in the classroom also in a different manner.
Thank you very much, sir.
Very nice.
Yeah, I'm a busy person. I was not very confident. I was going a little blank and I was thinking, oh, whether I'm saying it right or not. Okay, but I'm so glad to know that I've given a good presentation, sir.
It is perfect because students now will be reading books and teachers. But as a practicing pharmacist, when you come and present, then they realize that these are all things which normally being followed and they should be prepared for that.
Yes, yes, sir, definitely. In industry, there is a lot more learning, a lot more seriousness. Because I face PG audits, it's a very stressful time. We try to get things in order, in place, but when they come, they go non-stop. They write non-conformances, and if we just go blank and if we don't talk, yeah, Shruti doesn't understand some concepts. So we need to be very knowledgeable. We need to have a good knowledge about the regulatory requirements, and we need to implement in the company really well. And we need to have every minute details. We need to take care of it properly. So it's pretty serious when we go into industry.
Any more questions from the audience?
Any
Response?
There are no questions in the chat box, sir.
So shall we?
So anyway, the presentation was very simple. Use it and easy to understand.
Yeah,
Whatever they have seen, they have understood it because you have presented based on your own experience, and that makes a difference.
Yeah. I found the validation concepts to be very complex, but I tried to simplify and present so that people would understand it better and get connected better. Yeah.
Definitely, your efforts have paid.
Thank you, sir. Yeah. Thank you. I'll finish up my lecture by giving sincere thanks. Thank you, Subramaniam sir, for giving me this opportunity. It's really nice to be able to give this lecture. You have definitely inspired us quite a lot. I still remember I was in University, Swinburne University, and I was talking to my fellow colleagues and friends from India, from Punjab, Gujarat, different places. I just mentioned my principal sir is C.V.S. Subramaniam. They immediately said, "Oh, your principal sir is C.V.S. Subramaniam. We read the books from C.V.S. sir." It was a proud moment. You are the author of really lots of books, and you have done books really useful to all the people. So I'm really lucky to have you as my principal.
Thank you.
And thank you, sir, Kulkarni sir and Gangaraj sir, for convening this lecture. Thank you, sir, Gangaraj sir. It's nice to have you as my lecturer.
Yeah.
And you have supported me and you have encouraged me quite a lot in my career. Thank you very much, sir.
Shall we conclude, sir?
Yeah.
Yes,
Dheeraj,
sir.
sir.
Yes, sir.
Shall we conclude?
Yes, sir. We can conclude.
Oh, 1 minute. Let me see what is there in the master. What is
Some chart.
after
Some question
read,
has
sir.
come.
Yes. First one is thank you very much for attending webinar and making grand success. After doing masters from India, can we do a job in foreign country directly? After doing PG course, say for M.Com.
I know what you mean. That's a difficult question at the moment because in Australia, the PR regulations have got very strict. They have got only a list of occupations where they need more people. So for those occupations like IT, it's easier for them to come here, find a job, and get a permanent residence. But for our pharmaceuticals, they don't need much. So it's a little difficult. So you need to come here, talk to an employer, and work and get the employer sponsorship, which is a little difficult process, I could say. I have actually gone through the process because soon after I finished my master's, they have taken quality assurance course, pharmaceutical course away from the list of occupations what they require. But I'm so glad my ex-employer has liked my job and he has sponsored me. So thereafter I got the work visa, and then after I got the PR, and then after I got the citizenship.
Okay, thank you, ma'am.
Thank you. So since there are no more questions in the chat box, take this opportunity to thank Shruti Challa for sparing her valuable time with us and informing us about the actual position of pharmacist role in the industry. Thank you very much for sharing your thoughts with us. I also thank all the participants and staff and students and management for attending and getting the benefit from her experiences. Thank you very much, every one of you. Thank you.
Thank you,
Thank you
Shruti,
very much,
for
everyone.
your presence
Right.
Thank you, sir.
and your
Thank
valuable
you.
lecture.
Thank you very much, sir. Yeah, if you need me to do more lectures, maybe maybe
Yeah,
once
Definitely.
or sometime later, you can always feel free to approach me, but not too much because yeah, I'm a busy person myself. Now I got a
Yeah.
baby.
Yeah. But this time, with a short time, you have agreed for.
Thank you, sir. Thanks, sir. Yeah, yes, thank you very much, sir. Stay safe, everybody. Stay safe. Take care of your health. It's really nice. I'll be attending more alumni. It's really nice to see you all after so many years. And you have never changed. You look the same. Gangaraj sir look the same. You've been maintaining good health, which is really a good thing to see. Thank you very much. It's an absolute pleasure to be here. Have a good day.
All the
Thank
best
you.
today.
Have a good day.
Thank you,
Yeah,
sir.
all the best. Yeah.
Sir, previous sir. Thank you, everybody. I'm ending the session here. Bye.
Thank you.
We
Bye.
will leave the meeting, sir. Right.
You can get in touch with me if you want this PowerPoint presentation. Probably I'll send it to CVS sir and also Srinivas sir. Thank you, Srinivas sir, for doing this call yesterday and helping me with the IT requirements. CVS sir, Srinivas sir. Probably I'll be sharing the PowerPoint with Srinivas sir, and probably if any students are interested, they can take the PowerPoint. And I'll give you obviously my email ID and LinkedIn. So if anybody wants to get connected through LinkedIn, you can get in touch. Thank you very much. Have a good day.
Thank you.